Welcome back to our fourth video of the series where we talk about transcription, which I just realized I badly misspelled. There we go, transcription. This is the process of making RNA from DNA. DNA cannot leave the nucleus to give instructions. So if it wants to make a change in the cell, if it wants to make a protein, if something needs to happen in the cell, it needs to send out a message, which this RNA is. And specifically, this is the process of making mRNA from DNA. So by making mRNA, it is able to make a message and send it out to the cell so that the cell can receive it and make whatever needs to be made. Now, we will remember in RNA, we have 4 bases: adenine, uracil, cytosine, and guanine. Adenine always matches with uracil, and cytosine always matches with guanine, which means if this is on one side, this will be on the other. If this is on one side, this will be on the other. So we start by taking our double helix DNA and unzipping it so both sides of the code are exposed. When we have exposed both sides of the RNA or the DNA, so this is DNA. It is the template for how it is to be built. So we unzip the DNA and we build almost the same thing that we have in DNA when we built it. DNA had an enzyme called DNA polymerase. Now, to build the RNA, we have an RNA polymerase. And that comes along and reads the first letter. For this example, we're going to be using the bottom. So it reads the first letter is T. Therefore, in DNA, T matches with A. So it throws up in A. Now, next letter it reads is an A. Normally in DNA, we would throw a T here. But in RNA, there is no T. So when you see an A, we have to throw in a U. Goes along and finds the C. Realizes it needs to match a G. Another C, another G, a G. We have C, a G, C, T, A, and so forth. And it goes along and keeps matching up what letters need to be made in the RNA. When it reaches the end and the DNA tells it that is the size of the protein that needs to be made, it zips the DNA back up. Because just like when you are reading a recipe book, you have your recipe for what you need to make. When you're done with the recipe book, you close it up until you need to use it again. Same thing happens with your DNA. You have your recipe. You zip it back up, and you release the RNA off to go find the ribosome, where you will be able to make this protein using the recipe that is mRNA. And that is the process of transcription. With the next video, we will be looking at how you use this mRNA and turn the code that is on here into a protein that your body can use. And that is called translation.